There we go, record. Beautiful. All right. So don't worry about the formal intros because all the gubbins will be down below. But I was thinking, I think the last time I saw you was not in person. It was 2 years ago, 2.5 years ago at the Finding Hemo conference because you gave the keynote, right? In 2022. God, time flies.
Yes, all the Zoom-based meetings are kind of a blur for me. I'm like, what year was that?
Unbelievable, isn't it? Yeah, the pandemic's almost 5 years old. It occurred to me this morning. That's just so scary. Wow. Mm. So anyway, yeah, I don't know where you want to kick off. I've got a few questions I can fire your way, but what have you been up to? I know you sleep in CSF amongst bold. So what's your current focus?
Oh, yeah. I mean, I guess kind of along two lines. And one is, yeah, sleep and CSF. So we're really interested in finding better strategies to image the movement of CSF in the brain. And the goal is really to kind of understand what regulates it. And so sleep has been a big focus since it really seems to have a really profound effect on CSF dynamics in the brain. So we're interested both in kind of how can we make our methods more accurate to really understand what we're measuring about CSF flow, and then how could we use them um, to try to understand the control systems for this fluid and, eventually hoping to really understand the function, right? Why would it flow so much in one case and not in the other case? What would be like the physiological consequences of that? So I think kind of fluid flows in the brain is a main interest. And it's a really exciting one because it feels like there's a huge amount happening in this space. It's kind of taking off. There's even a new study group at the ISMRM on neurofluids of a community of people who are interested in that. And then I'd say the second kind of big thread of interest for us is, one that I've had for a while, which is how could we get more temporally precise information out of the fMRI signal, really to try to understand timing. And the application there is to begin to kind of use kind of temporally precise methods to start to try to understand sequences of things happening in the brain. And again, in the context of sleep, the idea is to kind of start to use timing to understand what happens first. So this could be, for example, in like the control circuits that regulate your sleep or wake state, using timing to understand chains of events, and also to kind of think about how more broadly could we analyze temporal information in fMRI in a way that could benefit neuroscience experiments.
Excellent. Okay, you're going to fall into my cunning trap. Cool. That's not quite true. Okay, that's really interesting. And then, yeah, on the CSF flow. So the fourth ventricle CSF flow dynamics you're using because you can see it, I presume, right? It's because it's the low-hanging fruit. It's not, obviously it's a step towards what you would like to do, which is to be able to look at CSF flux, CSF flow very locally subvoxel level, which of course right now is a real issue for us. I mean, there's people trying various things, but all of those methods seem to be either highly motion sensitive or highly insensitive in terms of signal to noise. And also, even then, there's still a leap of faith in terms of which compartments are really driving what you're seeing at the voxel level. So I don't know that anybody in the MRI world has has looked at this. So before I get a quick one, and then we can get back to MRI. Have you spoken to Larry about doing MPI for CSF?
No, I
Yeah.
haven't.
You need
Interesting
to,
question.
please.
Write that down.
Yeah, because it's positive contrast, and that's why I got Steve Conolly on this a few months ago. Larry, I'm hoping I can get Larry on in the next few weeks because I've always struggled to know what would be the killer app for MPI. Right? If you're just going to do functional MPI, you just got another vascular imaging method. Who cares? It's just vaso. It's just another expensive version of doing vaso, with probably very few spatial or temporal benefits. Okay, you get positive contrast, but that's it. But if you're going to bother to do intrathecal injections, etc., of spions or whatever, anyway, for MRI contrast, well, if you can move to positive contrast and actually and also have essentially an indefinite time course, because as these things stay active as MPI particles, they diffuse days, weeks, months. You don't care as long as there's enough signal there that you can measure them. You can measure that flux, and that might be the time course that we need to see CSF flow.
Yeah, that's such an interesting idea. There's very, very little work being done with intrathecal injections. There's like a group in Norway that's had some really interesting papers with that, but I hadn't made the connection, yeah, with Larry. So yeah, I'll definitely talk with him about that. That's a super interesting suggestion. I think the intrathecal injection has always seemed so um challenging to me that, yeah, we haven't kind of
Yeah, I
explored
think, putting a, I'm not a clinician, but if you have a clinical bent, if you're going, if you're just looking for interesting science, yeah, it's probably unjustified. But if you're talking about, if you had a reliable method
Yeah.
to characterize early onset Alzheimer's or hell's teeth,
Yeah.
that's a gimme, right? You're gonna do that every day of the week because you're going to do it once maybe, or once maybe every 5 years in a subject and
Yeah.
follow them, and that's probably an acceptable risk for many people if it's that serious of a condition.
Yeah.
I think the other thing is there's animal systems out there. It must be an MPI, animal MPI somewhere. So I'm sure a lot of this stuff could be done in rodents, and just give it a test, see what happens. Might be some really interesting stuff there for sleep dynamics too, right?
Yeah.
From all of the other stuff. So cool. All right. Well, cool. That's good. Um, so yeah. So going back to the CSF flow and my haha cunning trap. I've been talking to Simon Eichhoff and Vera Komeyer about confounds, pejorative term, and I think I've read your papers, your bioRxiv papers, especially that one where you predict the EEG signal from the fMRI in the
Mhm.
sleep, and that is just brilliant because first of all, it's model free, right? It's just information, so you're just trying to back predict what the EEG would have given rise to the fMRI, which is lovely. But you also with that paper and then the other one that you wrote, that I think is still a preprint, when you're looking at the CSF dynamics in relation to sleep staging as you go, or you've got sleep deprivation, I think, in that paper. You've got sleep deprivation and the awake and sleep deprived state, and people are making errors, and you see that there are these sympathetic responses and that there's an awful lot of predictive power in the respiration and the pupillometry, etc. And it's like, well, yes, that is exactly that's such an important insight because I think where we are in the field is we're still treating respiration as a confound, and we want to do the magic, wish it away, and then we'll start really working, and we'll talk about everything that's left. And I think that's a categorical philosophical mistake. So I really like the way that you're approaching this. So I have a question about the EEG fMRI specifically, because I was thinking about this when I read the paper. You did a retro I-core type correction for the respiration, I believe, before you did the EEG prediction. What if you'd left the respiration in there? What would have happened?
Yeah, so I think so firstly, it's funny that you call this a trap because I feel like I agree with everything you just said. So
Good.
it's actually really great to hear it because especially this concept of how physiological kind of like noise is handled sometimes in fMRI, I feel like really is missing a ton of information, right? It's like, yes, those signals are in there, but they're really meaningful signals. And also, if you just try to regress them out, you're probably actually distorting them in ways that leaves them in there in kind of a confusing way. And so, like in that paper, I think that actually things wouldn't necessarily have changed that much if we left those signals in. And the main reason is that we were actually trying to kind of account for this in the model in the sense that like we would leave in just ventricles and white matter and have those be predictors too. And so we were really often looking for predictive information beyond what's in a region that has kind of like less neuronal contribution or presumably no, although I hesitate to say that, neuronal contribution to the bold signal. So I don't think it would have hugely changed the results, but it's a great point. Like we could actually try it. We could try to do this prediction with or without. You can actually predict lots of interesting things just from systemic physiology, right? There's even a paper from someone else here at MIT where they just take people's respiration and they try to predict their sleep from their breathing alone. And it's really remarkably effective. So there's a lot of information there once we're in this world of kind of just using the information. So I think our strategy has tried to be like, can we see like what distinct non-overlapping information there is once you kind of take those other signals into account.
Okay. Yeah. I failed to spot that. Yeah, of course, because if the ventricular, most of what you see in the fourth ventricle is reflected from the lateral ventricles. So yeah, if you just make that's a shadow of whatever's going on in the systemic physiology
Exactly.
anyway, then
Yeah.
yes,
And that's
okay.
always there. Like there's
Yeah,
nothing
yeah,
you can
yeah.
actually do to get rid of it. Like no matter our data cleaning can be fairly effective, but you can't erase it. But one thing we found, which we actually expected, although I don't know if other people would have agreed, is that like just the CSF signal on its own was predictive of EEG delta waves. And that made sense to us because we actually already know that they're correlated. But it also means that like uh, you know, pieces of that signal could be anywhere. There's CSF around the entire cortex, right? And so we wanted to kind of put into our model that we had to find regions that were predictive beyond just the level we would have gotten a prediction from the CSF signal itself. And that also carries, yeah, the cardiac signals and the respiratory signals.
Yeah,
Yeah.
because when you look at just the fourth ventricle, that's it's essentially the integration of the entire brain. So it's going to be dominated by the whatever the volume average is, the biggest volume average.
Exactly. Yeah. Yeah.
So I suppose you separated the delta and alpha frequencies anyway in that nicely. So I suppose if there was, maybe you would end up with some sort of third type of network that might have been just mostly sort of vascular CSF. Maybe it's purely respiratory that's just being driven by blood gases, let's say, that had no other neuronal component, and it would only be localized. It would probably look like an angiogram, I would have thought, if there was a part of the brain that looked like that.
Yeah, I think the challenge though is that like even those changes in blood gases, for example, they're actually really centrally controlled and they do
Mhm.
co-agree with neuronal activity. And so there's this nice preprint from Katie Chang's group online right now, where, for example, they're looking at kind of this type of whole brain sort of dominant fluctuation that's kind of global. And one thing I thought was cool in their paper is they actually then clamped CO2. And
Mhm.
so they kind of could control for that by trying to stabilize the CO2 levels in the blood. And when they did that, the pattern was actually still there. And so, while of course CO2 fluctuations can cause these really large vascular effects, still that same pattern seems present even when you kind of fix that in place. And I think it partly is also that there's all of these neuromodulatory changes, like changes in arousal are causing changes in noradrenaline, which directly act on the blood vessels independently of any kind of like respiratory change itself. And so although you could sort of say it's purely vascular, at the end it might look pretty similar also to a neuronally regulated kind of central change, like not through local neurovascular coupling, but through the fact that the brain at the end of the day is actually kind of regulating global vasoconstriction as well.
Okay, that makes sense. So perhaps there might be a phase difference if you were able to seed, say, in the carotids and go forwards in time. If it doesn't flow at the rate of the blood, then it's probably not blood. But then again, if you ended up with sort of astrocytic, innervation that is primarily going in the direction of the blood flow, so it's dilating and it's cascading in a sort of like a gap junction down an axon was going fire, 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 fire. If it's doing that down upstream first because it's going to flow the blood downstream, because that's the smart way to, no point in dilating when you have nothing to dilate for, right? You want to dilate upstream first and then propagate that little bolus of blood down. Then it would look exactly the same, and the flow rate would be the same, the phase would be the same, the mechanism will be different, but you wouldn't be able to tell because it
Yeah.
would be the same in your blood dynamics. Yeah, that's really interesting. That's really
I mean,
interesting.
that's a great point that like, yeah, things could look really similar. One thing Daniel Gomez, who's in the lab, he kind of presented at ISMRM some work just trying to compare timing of signals driven by neurovascular coupling versus by a breath hold. And, some people actually try to use that, right, to correct fMRI, essentially to say, let
Mhm.
me use a map of cerebrovascular activity to get a sense of kind of my signal amplitude and then adjust for that when I'm doing voxel-wise analyses. And he kind of directly compared those at really high resolution. And I think for amplitude of signals, it works really well because, like you said, they're kind of very similar signals. But for timing, it worked really noticeably less well. And I think that reflects that neurovascular coupling, things tend to originate at the site of the neuronal activity, whereas for kind of vascular drive, they don't. It's sort of a different mechanism driving the change in flow. So I do wonder if like trying to exploit that timing information could be helpful for disambiguating kind of like which of the mechanisms is at
I
play
wonder
here.
if you might need a spatial as well as a temporal
Yeah.
domain. Where machine learning might figure this out. You can imagine if you have a centrally orchestrated brain-wide activity, then things are happening with one particular, let's say the timing is the same, but you have all of these disparate nodes in the brain that are all being orchestrated at the same time. With blood flow, it must follow the vessel structure. So if it so the spatial constraints are different, right? If
Mm.
it looks like a blood vessel, it's probably in the blood vessel location. It's probably blood-borne. But if it's outside of that, it can't. I don't think it can easily be explained as a blood-borne phenomenon. So maybe if you have a spatial as well as a temporal, that would allow you to
Yeah.
separate it. And I think that's the beautiful thing about that paper, your EEG fMRI paper. that it was model free, that those networks emerged from just simply the predictive power of the fMRI. Maybe something like that could be done with the timing information, and
Yeah.
it might tell you if it ends up looking like an angiogram. Okie dokie. Right, all right.
Yeah.
Because we can map that, right? We can get ground truth for that. It's pretty easy. Okay, well that's super cool. All right, well we got straight into the weeds, didn't we? So I have another quick question just to make sure that I understand what because the the glymphatic system confuses me. I think I kind of understand it. I think the part, but the difference between the classic view of CSF flow and lymphatics is just that you have the virtual Robin spaces, you have the CSF coming in along the peri-arterial spaces. That's known, that's classic physiology. But what's different is then you have the aquaporin channels, the route through the tissue, and then it vents through perivenous spaces. So I guess the sort of the new part is the perivenous spaces and that route through, right? That you've got aquaporin channels and you go through. So what is your understanding, when we're looking at the fourth ventricle dynamics, and we talk about that as reflecting CSF flow, or if you're doing intrathecal injections and then it's going to do something interesting in the brain, is your assumption then that the CSF, the route of CSF into the brain is from the choroid plexus, but lateral ventricles, fourth ventricle, back around through the meninges, and then back in, re-entrant down these virtual Robin spaces, and that's how we then start to get the real dynamics of CSF.
Can you say your proposed route? Sorry, can you say it
So,
again?
because classically we know
Yeah.
CSF comes from choroid plexus, and then we would, and if you look at the physiology literature, there's sort of arrows go, third, lateral ventricles, third, fourth,
Yeah.
and then they sort of just put arrows on, and then they
Yeah.
just sort of say, well, some stuff goes this way around the spine, some goes around the head, and then it disappears somehow. Magic happens.
Yeah.
But then I think what we're saying is, and the reason why the intrathecal route must work is because what we're saying is, well, the fourth ventricle is doing its thing. It's a pop-off valve for the brain-wide dynamics. But what's important is that the CSF, once it's created and come out of the middle of the brain, then is around in the sulci, and it can re-enter the brain into the ISF because it's coming in through these periarterial spaces. Is that what your understanding is?
So I guess, I mean, I think what my understanding is, I've definitely there's these textbook cartoons and I use them in my talks. I think they're really helpful for kind of getting a sense of the anatomy of the space. And certainly they've always come with these arrows of the direction of the flow. I think that what they're trying to represent there is kind of a net flow, right? It's that we're always making CSF, we're always draining CSF. That happens a few times a day. And so there's this net movement from the choroid plexus to whatever the sites of drainage are. And, I think it's clear that there's multiple sites of drainage and not just one. And I think that's actually one of the really fascinating aspects of the glymphatic model is that, it's still very new. It's only been around for maybe 10 years. And there's just super interesting papers coming out all the time on different exit routes and different circulation pathways. But what I'd say is that those arrows are representing net flow of something that's happening maybe 3 times a day. So it's really, really slow. And I think that what we're looking at is totally different because it's pulsatile dynamics on the time scale of seconds. And those pulsatile dynamics are huge. They're clearly corresponding to fluids flowing at multiple centimeters per second. So I think that riding on top of the slow net drainage is a much more rapid pulsing process. And so I think because of that, any model you would make based on just that slow CSF production and movement is not going to capture all what actually happens to transport when things are moving really fast up and then actually really fast down again, right? And so I think that the advent of all these tracer studies is really exciting. And what would be great is to actually combine it with computational fluid dynamics models that use those pulsatile dynamics to understand how much of this is making more CSF and gradually draining it, and how much of it is actually these oscillating waves that are kind of pushing things at much faster velocities than you would otherwise get. Does that sort of answer your question? Because I feel
Okay.
for example, it could have that trajectory for sure for CSF, but based on what we're measuring in the ventricle, I think it's also really clear that At a faster time scale, things are moving many, many centimeters back and forth. And as you said, we started by measuring in the fourth ventricle, but because the signal is great. It's like we can see it. It's clearly a velocity signal. It's very sensitive. We have good temporal resolution, but it's a very coarse readout. We kind of only know macroscopically at one point at this place that's kind of like integrating everything that's happening over the brain. And so we can't really make an assumption yet for like what we're measuring there, what would mean is happening like in the tissue or around the tissue. That's what we kind of want to do in the next wave, is sort of figure out like, okay, when I measure something like this at the ventricle, what can I infer? Is this like a coordinated action across many perivascular spaces simultaneously? Probably sometimes it is, and sometimes it isn't, kind of depending on the state of the brain. You could imagine basically anything that changes blood volume will have to change the CSF flow. And so whether that's a kind of coordination of neural activity, or like you said, just like pure respiratory effects, you can get beautiful CSF flow drive just from a breath hold. And so I think that depending on the drive of it, you're going to have really different things happening at the micro scale that might look pretty similar at that sort of ventricle level macro scale.
Yes, and I think that for me that would be the crux, because I can imagine the dynamics at the fourth ventricle as just a mechanical sort of pop-off valve, Monroe-Kelly doctrine. Great, that's one role. But that doesn't necessarily tell us anything about that 3 times a day wash cycle, which is what you're probably really interested in the sleep. What's going on at the tissue level, at the ISF level, and then where do the arrows point? What does the bulk flow look like at that microscopic scale? I mean, certainly, the tracer studies suggest that it is certainly reentrant. At least, yes, I think that's probably the case. If there are other pathways, they may or may not matter, I suppose. But as long as we've got at least one, right? The brain needs at least one.
Yes.
And I suppose the other thing is just to keep in mind, as you were saying, describing that, as like, we keep wanting to ascribe single functions to things, and it doesn't make sense. I mean, the blood flow, you could describe the blood flow as delivering glucose and oxygen. Well, but it also clears CO2, right? You can't do one without the other. That'd be kind of pointless, right? So maybe we just need to be a little bit more circumspect about multiple roles for CSF. And sometimes it's a pop-off valve. It's just a non-compressible fluid, and sometimes it's a wash rinse cycle that's going through the brain, and both are important, right? And you can't survive with one or the other. You need both. So okay, well that's cool. On that note, yes, I've been thinking a lot about the CO2 sensitivity. And one thing that I find, so there's if you haven't seen it, I'll send it to you. There's a review I read about a decade ago. It was written about a decade ago about CO2 sensitivity. There's people now looking in with anesthesia regimens, different anesthesia regimens that have induce different sensitivities to carbon dioxide in certain nuclei. And the review I read was talking about certain sort of essentially a CO2 sensitivity in like locus coeruleus and certain other nuclei, that they are the ones that they're the traffic police, that they need a certain level of CO2 to get you from awake to NREM sleep, but too much and it wakes you back up, because breathe faster, animal, or you die. But then that's a transition, a sort of a gateway into REM sleep, and that then your minute ventilation goes up again. But you need to go through that pathway. You can't just get there directly from awake to REM sleep. You need to go through the NREM sleep stage, and that's CO2 mediated. So one thing that I've been wondering about, and I thought about this the moment I read the Faults paper, the very first was, what's the mechanism? Because there didn't seem to be a way in your first paper to differentiate between what would be the respiratory, the CO2 response to causing vasodilation, and a brain-wide concerted activity. Do you have any better sense now about whether there's a crossover going on? Are they both happening with the same sensitivity, whether you're awake or asleep? What's going on with CO2?
Yeah, that's such a great question. So, firstly, I'd love to see that review article you mentioned. So please send it along later. So I think basically, sort of as I kind of alluded to, I do think that just both of those, like both things can affect CSF flow, undoubtedly, right? So I think that if anything, for a long time, it's been thought of that it's really, well, firstly, that the cardiac cycle was dominating CSF flow, but then that kind of respiration is the key driver, both through the actual pressure from the lungs and then also from these CO2 effects where you're kind of changing vasodilation. So I think, in some sense, that's kind of been a fairly well-established view, and I wouldn't contradict it at all. I think it's like very correct, right? That CO2 changes can drive changes in CSF flow. But I think that the idea that like neurons can actually regulate this is a really fascinating one because it makes the system become adaptive, right? It could be that the brain now could sense something about its environment and actually regulate its own local fluid environment, regulate essentially its own bath through that sensing. And so I do feel pretty certain now that there is this like important contribution of neuronal activity as well. And partly that's from this follow-up study that we did where we tried to kind of just do a proof of concept with visual stimulation. And there we kind of recorded respiration, we recorded cardiac function to make sure that we could do a kind of controlled setting where those were matched. And we designed a visual stimulus to just drive as much neural activity as we could. So it was like a pretty unpleasant flickering checkerboard. And even so, it drives much, much less synchronous neural activity than anything you would see in a change in arousal state, which is very global and very synchronous. But still, we're very visual creatures. So you can drive quite a bit of neural activity through an intense visual stimulus. So Stephanie Williams, a student in the lab, she led this paper. And it was very clear from her data that, you do get really robust CSF flow actually driven by local neurovascular coupling as well through the induction of the visual stimulus. And that's, I think, a nice case where definitely, I think CO2 can drive this, but neural activity alone can also do it. And it's funny because I think at first we were surprised by this and others were surprised by this, but then in retrospect, it's one of those things where you're like, well, how could it be any other way? Because we know we can drive changes in cerebral blood volume with neural activity. So how could it not drive CSF flow? And I thought what was nice was Steph was able to kind of create this model where she tried to predict the CSF flow just from the EEG in this case. And she didn't fit any parameters. She just assumed it was solely mediated by neurovascular coupling using the models we already know. And that model did really well at predicting the CSF flow. So basically, I think that both are there. And then the question is like, well, what's happening in sleep? And I think essentially in sleep, both are contributing. And that's actually very interesting because it suggests that if what you're interested in is what happens when something goes wrong with CSF flow, that there's actually two parallel pathways. And you could imagine now, like maybe if there's a disruption of the sleep itself or of these kinds of like vascular and respiratory dependent pathways, either of them could change the CSF flow. So I think kind of keeping that in mind is actually really useful for understanding at the individual level what these flow dynamics look like. So what we do now, which we weren't doing in the first paper, is now we actually always record end-tidal CO2 so that we can actually take that into account for every person. I think the real challenge is how do you analyze the data? Because it is controlled by arousal state, it co-varies with it, and it's collinear with it. And so what we're thinking about now is like, what's the right model design or analysis to kind of try to disentangle these two things, given that they really often go together. And to my mind, actually, the really interesting question is like, well, why do they go together? Why should your sleep cause this transformation of like breathing patterns in this way? Like, what's the functional purpose of that? Definitely the latter one you mentioned, right? If like CO2 gets too high, then we have to wake up, and that's the basis of sleep apnea, right? People who are kind of having these awakenings from sleep because their brain can sense that oxygen is too low. But why kind of in healthy sleep without these apneic events? This is kind of always oscillating. I'm very curious about that. That I don't know the answer to.
Yeah, which might be, that's this hypothesis in this review paper. I think that they're saying is that the sort of sleep center nuclei, locus coeruleus and others, require the CO2 to go up in order to basically get you in, so that they're gatekeeping, and these neurons are REM on, REM off neurons. And in order to get to, or NREM on, NREM off, and then REM on, REM off, to get to from one stage to the next, they're using CO2 and the chemoreflex as a way of sort of telling the organism what to do. So in a way, and it's also the switch to come back out of sleep, because you also not you need to get into sleep, you need a mechanism to come back out, right?
Yeah.
So I could think maybe CO2 is a fairly simple switch. The other thing that I've been thinking about too is if you did want to go from a state where you were blood gas dominant, let's say, when you're awake behaving, where you're changing respiration and that's making big changes. If the assumption is that you want to get this on the microscopic level, you want some kind of astrocytic coordinated activity to flux, to open aquaporin channels and cause local sort of modulation of vasomotion or whatever to drive CSF flow. And if that's what's the sort of the point here, at least one of the physiologic consequences of wanting to get into that state, then a fairly easy way to do this, at least in the cortex, is to just buffer it. Because if it's pH mediated, all you have to do is have those same neurons as they go into the sort of sleepy state, and they're all being brain-wide orchestrated. They just simply release a chemical to say, well, shift the sensitivity curve on the CO2. So as the CO2 level in the blood changes, we don't care. We're just going to ignore that for now, or we'll dumb it down, and instead we're going to listen to the music being played by the central orchestra. And so there's a fairly simple physiologic mechanism, I think. It's just a bit of biochemistry. And that would leave, and if that only happens in parts of the brain, let's say the cortex, if those subcortical neurons that are responsible for making you sleep, go into REM sleep and wake up, they can maintain that CO2 sensitivity. So they got to keep a hand on the pulse, right? We need to know what's going on in the blood, and we will tell the rest of the brain when to wake up and start paying attention to the CO2 level again. So I think there's some simple stuff there. Again, presupposes that we are correct in understanding what the brain is trying to do in the CSF flux locally. But I don't know it's necessarily important. I don't know. It may not matter. The CSF is presumably important. There's a rinse cycle going on somehow, somewhere, whether that cartoon is right or not. So the prediction there would be, if we could measure the ISF pH, it would change. Or maybe, yes, it would change, because it would have to, if the buffering of, if the CO2 sensitivity is pH mediated, then the ISF would have to essentially buffer that change. So the fact that the CO2 is coming in and there's carbonic acid, it's like, ignore, stay nearer to neutral pH, don't acidify, because otherwise it'll trigger vasodilation. So you turn that piece off, dumb that bit down, and maintain the astrocytic or the neurovascular coupling part. So I think there are a few predictions we could make. I'd have to look and see if we can measure the pH. We should be able to do that in spectroscopy.
Yeah, I mean, I think a rodent model would be the easiest way to do that, where you could really, yeah,
Has
easily.
anyone looked at pH with sleep?
Oh gosh, maybe. I'm not sure actually. I'll
Yeah.
look it up now, yeah, but nothing comes to mind, but it's such a huge field, it certainly could have been done, and I don't know.
Okay.
Yeah.
Okay. All right. Well, I'll think more about that. That's super important. Cool. All right. Um, so yeah, going back to what you said about processing methods, so first let me just applaud you for measuring more things, because I think that might be the one blind spot in the field. I think we just seem to have this MRI centric view, and we need to be measuring lots and lots of physiology, pupillometry. You've got end-tidal CO2. The more you can measure, the more you can then try to understand what the whole organism is doing in these different states. That's probably true of every type of fMRI experiment. It's got to be true of naturalistic viewing. It's got to be true of sleep studies. Why wouldn't it be true of most other things? Anything that's emotional, right? I mean, there's huge sympathetic responses. So what do you think then about the methods? Do we need a wholesale change? Do you think we're doing rings, the old sort of, GLM type models just need to be essentially retired, and we need to move to more data-driven, more AI type models, just with explanatory power, be more agnostic?
I don't. So firstly, I mean, I certainly agree with you that I think that these effects are kind of everywhere. I love studying sleep because I think it's fascinating, but also because I think it's a really interesting kind of extreme example of something that's happening all the time, which is that we're always tuning our arousal state. And wakefulness to sleep is easy for us to tell, but in many tasks, your arousal is fluctuating as your attention is shifting or your mood is shifting. So I think it's really powerful to take it into account. I wouldn't advocate for throwing out the GLM. I think the GLM is really useful. And I think that it's smart to take advantage of the new kind of machine learning methods that are coming out, but I think that also needs to be done in a thoughtful way, because it can be very difficult to interpret for the results of things like this. And ultimately, my goal isn't just to perfectly predict our data, it's to understand why it is that way. And so I think that, one of the challenges for us has been how do we take advantage of ML in such a way that we still have interpretation at the end of the day that lets us understand the system. So I feel like it would be great to kind of increase the community of people who are measuring these signals, but we do have to, yeah, we have to think really carefully about what do we do with that information now. And I feel like maybe it could be like GLM plus. Like I don't think these signals should just be regressors. We shouldn't just delete them. Maybe we could take advantage of AI strategies for figuring out like what information is in them. But I would love to also build more models that share the spirit of the GLM, where it's kind of like model based and we're trying to come up with linking functions, but that we don't just have one canonical hemodynamic response. Like instead, maybe if we really understood the relationship between these signals and spatiotemporal patterns that arise in the brain, maybe we could use that and not even just regress them out, but they actually carry really interesting information. So if you have a complex cognitive task, maybe actually you could use this to understand like what aspect of the response to my task is mediated by a kind of widespread neuromodulatory attention state change versus what reflects really kind of local cortical processing. So I would hope that we could kind of build up a toolbox to help people start to ask those kinds of questions. I do think it means that there's certain kinds of analyses that are very challenging to interpret in this context. So if you're just doing a functional connectivity analysis and you're getting a metric that's just a correlation, if, you know, your autonomic physiology is changing, that can change that correlation number for all number of reasons that are really hard to interpret. But if you kind of shift that to say, instead of just getting one connectivity metric, maybe I try to kind of analyze both of these components and understand how they are or are not present in my different individuals, you might be able to see all kinds of things that you couldn't see in the first analysis. So I feel like they're great to include, not just to rule out confounds, but to actually look at like, well, what is the neural process that's generating those? Why was it engaged by my task? And how might that reflect that actually, you know, I didn't process every trial the same way in the task. I was in a different attentional state in different ones, and that helped shape the kind of downstream responses.
So what I'm hearing is that you're advocating essentially indirectly for doing some kind of arousal-based measure of all these experiments, at least pupillometry or something EEG, as well as fMRI.
Yeah, I mean, EEG is so tough. So I definitely, I know that not everybody wants to put an EEG cap setup on before their fMRI experiment, but I think that's a great statement that maybe pupillometry is a nice intermediate where actually it's much easier to do than EEG, although it's still a bit of a pain. And there's a huge amount of information in the pupil. And so I think that that could be really interesting. I'm sure there's many people doing tasks where this isn't an issue. So if you have a really hard task and people are engaged all the time and you're only keeping the trials where they're doing it correctly, you've probably stabilized arousal. So I think it's more, this is,
Essentially subtracted it out at that point, right?
yeah, exactly. It's no longer varying in your
Yeah,
experiment. So
yeah,
it's no longer influencing things.
yeah.
Um, but I think that there's many experiments where it could be really interesting to add this dimension that, people's sort of energy levels are fluctuating over the course of the scan. And so,
And even if you only look at error trials,
yeah,
then at least you would have an explanatory power, like why did the person miss 1 in 10? And
yeah,
like, oh, look, because
yeah,
they zoned out, they were thinking about whatever.
yeah,
And then you would have that information. If you don't have that independent information, unless and until we have data-driven methods, in other words, we need lots of history, lots of validated fMRI experiments to then pull that out of the data. And until we have that,
yeah.
we need independent non-MRI methods, surely.
Yeah. But to your point, I mean, it is a good point that it is in the data. So for example, there was this paper from Peter Panettine's lab, like after we did that sleep CSF study, they just went into the HCP data and looked to see if they could find CSF signals there too. And they did, and they found it was actually really correlated with vigilance in the HCP data set. So actually, maybe even without adding other measures, maybe the same brain signals we've been measuring, they are in more conventional acquisitions. And so it's possible that there's actually enough in there to kind of get it out in more data-driven ways, like you're
Well, to go
suggesting.
back to what you said about, not treating the white matter and the CSF signals as just, junk, if the CSF signal might be your attention modulator signal.
Yeah. Yeah, maybe it's a helpful proxy for certain kinds of
Don't regress it
analyses.
out because you
Yeah.
just
Yeah.
oh, that's a really important point. That's a really interesting point. Oh boy, yeah, yeah, yeah. Okay, so maybe if we are going to keep the GLM, maybe we just need to rethink what we do with regressors of no interest, whatever that means.
Yeah, yeah, or
No
maybe
interest to whom?
yeah, exactly. Or maybe you kind of have a new maybe you kind of can split your high and low arousal things and try to model
Oh,
them
there we
separately.
go. Yeah, yeah. At the very least, yes. Or errors and no errors. Yeah, at the very least, to sort of do a sanity check come back. That's really cool. Okay, that's neat. I like that. So yeah, so because yes, as you say, EEG car, that's a lot of work.
Yeah, no, EG, I love EG fMRI. It's been such rich data, but I certainly like, I think it would slow the pace of progress immensely if everybody had to do them together. And for many, many studies, you don't need to do them together.
Do you think is there a skinny version? I mean, how many electrodes do you do on your? Do you do the full like 20 electrode set?
I think that that's such a great point. So we have 32 channel caps right
Mhm.
now, and we have to prep all of them because it's not
Yeah,
safe. Because you always have to have kind of low impedances in the scanner for
yeah,
safety and heating
yeah.
reasons. And I'm not aware of anyone selling an MR compatible EEG system that's just a couple electrodes. But you're completely right that if all you want to do is track arousal, actually just a handful of electrodes would be completely fine. So maybe somebody just needs to build that like, yeah, slim system.
If you just took skinny down your, if the electrodes are in the cap, then you have to worry about them. What if you just simply made a 4 electrode or an 8 electrode cap, and then it just blanked the other channels?
Yes, yeah, exactly. I mean, the problem is because for us, the amplifier goes into the magnet. Yeah, we would
Oh,
need
I
a whole
see. I see.
hardware
So you need to,
system.
okay. So
Yeah.
you need to
So
change that.
basically, it's very doable in theory.
Yeah.
It's more just a practical challenge, I think, of like of finding a manufacturer and a whole
Oh,
system.
Jesus, we just gave them a market. What are you kidding me? Yeah, they could sell
Yeah,
thousands of these
many
things.
people use this. Yeah,
They could
yeah.
sell dozens of 32 channels, or they could sell thousands of 4 channel systems. There we go. My goodness.
That sounds great to me. I would buy one. So,
Yeah, especially if it's easy setup. I mean, if it's kind of, if it's
yeah.
relatively simple, just a few electrodes, onboard gel, I don't know, some kind of little
Yeah.
self-gelling system so you don't have to keep futzing with it and all the rest of it. That would be super, super cool. And then just at least get attention monitoring out of it so that you have that independent, maybe even do sleep staging. Is there a minimum number of electrodes you need to do sleep staging?
I think the ASM suggests, for example, like 6 as the
Okay.
minimum, but in reality, yeah, you can get a ton of information out of like 2.
Okay. Okay,
So,
cool. So there we go. Well, people listening, so you can create a whole new market for us. That's super interesting. And then do you guys do pupillometry as well as EEG anyway? Yes.
so we try to. So it's really easy to turn on the camera and record video of the eye, and you get eye open, eye closed very
Okay.
easily. What's a little more work is to actually get pupil diameter because then you have to do these calibrations so you can try to actually estimate the diameter of the pupil as it's moving. So that we don't get routinely because it just there's kind of more prep cost time, but we do it sometimes. Yeah.
Okay. Okay. All right. Well, that's really
I think
interesting.
even eyes open closed for resting state studies is still very helpful.
Yeah.
Knowing
I mean,
people close their eyes, I think is pretty useful.
yeah, yeah. I've never done it, but I've watched people setting it up, and you see people, their eyes glaze, and they see the slight slow saccade or whatever. Yeah, you
Yeah.
can tell when people are getting drowsy. It's
Yeah,
pretty
it's funny because with
remarkable.
these sleep studies, people so often ask me like, how do you get people to fall asleep in the scanner? That's half the challenge is like, how do you keep people
Ah,
awake in the
yeah,
scanner?
yeah. Have they never done fMRI? I mean, it's a cure for insomnia. It's the most soporific device ever invented. Yeah, the
I
people
think
fight.
yeah,
Yeah, yeah. Like
there's a lot of
one,
individual differences. I mean, some people are very alert in there,
yeah, yeah,
but yeah,
yeah.
a lot of people fall.
1:30 in the afternoon after lunch. You kidding me? You're gonna pay me to take a nap in the scanner? That's great. Fantastic. No, that's yeah, yeah, absolutely. I think the arousal thing is really important. Do you do much on the diurnal side then? Separate from sleep awake, have you looked, in regular, well-rested people look at early in the morning and what happens
Wow.
during the day?
Like circadian rhythms, I
Hmm.
haven't, although I think it's absolutely fascinating. So for now, we've basically just tried to control for that. So all of our studies rule control for the circadian effect. So when we look at sleep and wakefulness, it's at the same time of day. So everything's kind of accounted for. And then in our daytime studies too, if we can do same time of day, sleep versus wake, or we manipulate people's sleep. So in some sense, in the data we've collected, it's actually impossible to look at that because we've controlled for it. But there's clearly really interesting time of day effects in fMRI. I think Thomas Yeo has a paper where they looked at kind of even just the amplitude of fMRI signals over the course of the day, and they changed quite a bit. So I think it's a really interesting area to go into, and I have been thinking a lot lately about a dense sampling study to try to kind of look at those factors. But it's also tough because everything is changing with time of day. So when we do like sleep versus wake at the same time of day, we can control it, the circadian effect, and just look at state. But you can't do that when you're just scanning someone over the course of the day because the time is changing, but also their arousal state is changing because they are getting more or less tired. And then also things like their hydration and, all of these other factors are also cycling with time of day. So I think it's a fascinating question, and I'm sure it impacts this a lot, but I haven't looked at it personally.
Maybe, yeah, maybe looking at the insulin swing, maybe looking at the pre-post prandial effects. That might be interesting too. It's another state that you can manipulate fairly reliably in
Yeah.
most people, like sleep, and it's more sort of a bistable state. Because I like what you said about using sleep as a way of, it's a nice sort of slow, predictable way of putting people into known states, which with emotional stimuli, much harder, right? They're
Yeah.
much more fleeting, which but the postprandial state, that's pretty long lived. It's going to be tens of minutes, hours.
Yeah.
So that could
And
be
the animal work has looked at circadian systems quite a bit for things like just brain clearance and things like that. And they do find strong effects of circadian time, both on clearance and also on like the brain immune system. And so like macrophages and things like that. So
interesting. So on that particular note, so I'm trying to write a review, sort of a review. It's a summary of all of the confounds. In other words, it's not really, that's the term that everybody uses, right? It's really just biological routes into imprinting signatures on fMRI time series data. All the possible forward feedback ways that you can end up with information in the data that you didn't necessarily expect. Like the weight is the classic one, right? People were surprised when body weight got encoded as a phenotype, right? And then it turns
okay,
out,
interesting.
remember
Yeah.
that one because of respiration, because of the size of chest, and the pseudo motion. But there's also mechanical resonances in the bed, and there's other routes, right? There's lots of ways that biology can sneak in through the back door and you don't realize it. But the one that comes in through the front door that we all conveniently neglect most of the time is caffeine. And when I got to that stage, it took me days to get through that section. Um, so what do you do for caffeine? Because there doesn't seem to be, you're damned if you do, you're damned if you don't. If you're caffeine, 80% of your subjects take caffeine. If you tell them to stop, their adenosine level goes up, the receptors are more numerous anyway. You get a different physiologic state than they are normally. But you give them coffee, then what?
I know. I totally agonized over this. Yeah.
Yeah,
Especially because in sleep studies, it's really common to just say, okay, just stop caffeine. But I mean, if you
it's
ask
an
me,
abnormal
not have
state.
coffee, it's
Yeah.
not normal. So I've wrestled with this a lot. We've come to different solutions for different projects. One that we like to take, for example, if it's a sleep study, is firstly just enrolling people who don't consume much caffeine in the first place. So we have baseline caffeine consumption as an exclusion criteria. And so somebody who's having, several coffees a day is going to be excluded from certain studies that
How do
are
you assess that? Is this just
just
self-report?
self-report, just
Okay.
self-report.
Because
Yeah.
there's that website which I'll forward to you. I'll put it in the links too. There's a website where you can track inadvertent routes from caffeine. It's amazing how much caffeine you can get without thinking you drink coffee, chocolates, all
Yeah.
kinds of different sources, even without doing the energy drinks. Yeah, there's all kinds
Yeah.
of backdoor routes that you can get a lot of caffeine. But carry on.
Yeah. Well, I'd like to see that one. That's a good point. But we do go to some degree on self-report. And then other than that, right now, we've been just taking kind of within subject designs as a strategy. So although some people may be in slight caffeine withdrawal, whereas others are not, the comparisons that we're doing are within subject.
Yeah.
So they're matched across condition for the caffeine state that they're in. So that's how we mostly handle it for arousal studies. But
Density
this
rate.
is an issue for all fMRI studies because it's such a
Yeah.
potent is a vascular modulator that it would be nice if we would all start. It would be actually a cool community effort to have like some scale we agree on like how would we do the reporting of this and at least just log it so that we know for
That's what I think. Just treat it like any other physiologic parameter. If we just measure it, at least we can look at its explanatory power. Because I think either telling somebody to stop who normally uses, or the alternative, which is to spike everybody,
yeah.
right? Like clamping, like with CO2, right? We're going to give you all a triple espresso. Too
Yeah, I
bad,
wouldn't do that one.
right? Yeah, everyone's going to be jacked. Either way, it's an approach. But yeah, as you say, damned if you do, damned if you don't. But if we measure it at least, or try to assess it, maybe a little bit less than self-report, maybe at least try to gauge it,
Yeah.
then it would be like body weight or another parameter that is a sort of a rough proxy for what can be sort of a trait-like phenomenon, right? I think that's kind of how I've been thinking about caffeine. It's like cardiovascular fitness level. It's state-like right now, but it's also trait-like. You can change it. If somebody decides to go through an epiphany and starts exercising, they will change their cardiovascular fitness. And if you don't record that, then you're not going to know why their brains look different.
Yep.
And I think that's sort of, I think, how we should be at least approaching caffeine. If we don't measure, at least we should be measuring it. Then we can figure out whether it really matters or not. And it probably does. I like the within subjects approach. That's a good one. So when you're doing these time of day, when you're doing these sleep studies, what is the time then that people are being tested?
Yeah, usually at midnight.
At midnight, and then in the sleep deprived state as well at the following
Oh,
midnight?
so yeah. So when we're doing just a pure sleep study, we're usually scanning around midnight. We're kind of trying to start a scan around somebody's natural bedtime and then go on
Okay.
from there. So it's easy to have them not have had caffeine recently because a
Yeah.
lot of people only have it in the morning
Yeah.
anyway. So we're at least,
Half-life's like an hour or two, right? So if they've had it even at 3 in the afternoon, like me, then it's pretty much gone, you assume, by midnight.
I wouldn't assume it's gone. I think the half-life is about maybe 8 hours,
Oh, is
but
it really? Okay, that long.
yeah, but I think certainly like if it's in the morning, I think it's pretty well cleared. And we
Okay,
usually do have a constraint that you can't have caffeine after noon before coming in
gotcha.
for an
Okay.
study. So I think for those people, I think it's probably at pretty low levels already, and we're doing matched. For when we looked at sleep deprivation, we did the scans in the morning. And so in that case, it was matched within subject that it was in the morning with no caffeine. But in that case, it would have been harder for people. It would have had to be like try to scan them before and, when they're not very caffeine deprived yet, because they're maybe only an hour or two late for their normal morning cup, and then they can get it after the scan instead.
Speak for yourself. I can't get out of bed without coffee.
I'm actually famous for my inability to function without coffee. It usually results in me like making a terrible mistake while I'm making the coffee. It spills everywhere, and I'm like, oh, how do you make coffee before you've had
The
coffee?
grounds where the water's supposed to go. I've done that before.
Exactly. Yeah.
Yeah, yeah, yeah. Um, I was thinking about there was something else you said that was triggering a thought about the caffeinated state. Yeah. Okay. So you're controlling for at least the time of day and the caffeine level at
Yeah,
the
it's controlled for, but I love your suggestion that this is some info like we already it's so common in MR to gather handedness information, right? It would be really cool to just add this to that as an
yeah.
extra thing to be aware of.
Yeah, one of the points of this paper, I've been because again, I've been talking a lot to Simon Eichhoff, and he's trying to do precision medicine with his ML. And I have some ideas of ways we can use caffeine, perhaps as a sort of a cartoon system to try to because it's so common and you can easily manipulate it. It makes sense if you're going to build models because you can get richer data sets, labeled data, right? If you're looking at disease states, you get what you get. It's
Yeah.
very slow, very long, right? The N is always going to be small, so it's a bad place to start. Um, but yeah, thinking about what we collect, I've noticed that in psychology labs, handedness, language abilities, education level, all standard. Then
Mhm.
you look at studies that are being uploaded to databases from hospitals, weight, height, maybe blood glucose, genetics, right? But no handedness, no language, no education level, no demographics. So, and it's just like, well, hang on, these are opposites, right? We've got at least got to have some sort of shared, common set of medical things that psychologists collect, and vice versa, because otherwise we're going to end up with this bifurcated data library of data out there that may or may not be that useful, right? Because it's going to be very, very poor in certain dimensions of information based on whether it's medical data or scientific data. That's going to potentially be a problem. I mean, like from my place, we don't even do accurate body weight in the header. We just guesstimate it, put it in. Right? That's pretty shit. We should probably fix that, right? We should probably at least have a scale. We do have a scale. Nobody uses it, right?
Yeah.
But we should probably start getting that accurately. But I don't know what other people do. But we should probably start to agree on minimum collection of things.
Yeah. It's a tough balance because I think it's really important to have all of this for really big data, right? Like
Yeah.
but at the same time, when you're running a study in 8 people, it's actually maybe burdensome to collect every personal piece of information about that person. And you're never going to be able to actually draw a statistically justified conclusion from an N that's small about these kinds of demographic factors. So yeah, it is a kind of challenge. I would love ways that make it like easy to do,
Yeah,
right? So that there could be like community buy-in, that this is like a low cost incentive.
yeah.
Yeah.
Yeah, I mean, a way to do it would be a central sort of web-based system, right? Where anyone who's doing an fMRI study, the subject comes on and does it themselves in the 20 minutes before the scan, and everybody's using the same system, and it's just logged. You get an alphanumeric reference, and you can either use
Yeah.
that to register so that you can tie the responses to the data if you want. If not, for privacy concerns, you use you air gap it,
Yeah.
and then have a key that's kept somewhere else. But that sounds doable. That's the sort of thing that could be done at the OHBM level, you would think.
I love that suggestion because yeah, then it's being provided as a service, and I think if people
Yeah,
are way more likely to use it in that
yeah,
case, that's
yeah.
great.
I'll mention that too to Emily Jacob because she's doing the Women's Brain Health Initiative, and it's very organic, right? They're just making things work as they go along. They don't have any real they have goals, but not like pathways to get there. They're like, for example, right now they're just taking the data that people offer them,
Mhm.
structurals at least. Eventually, they will probably ask, can you, if you don't in your protocol include an ASL or whatever, then please if you're going to do it, please select from this little library of scan types because these are validated. This would suit our
Okay.
criteria. If you've got your own going in and it's good enough, okay, fine, we'll take that because something better than nothing. But that's the sort of thing I think that they would like because they need phenotypes, they want blood draws. Everything needs to be very simple if you're going to scale this.
Yeah.
So I'll mention it to her. Maybe that's something that we can do through the WBHI and then roll it out
Yeah,
um,
that'd be
from,
amazing.
from there. That would be cool. All right, I'll mention that. All right, what else? What else, kid?
Well, I should
Yeah,
probably wrap up.
yeah, you got to get out of here. Yeah, it's 11 o'clock, 2 o'clock your time. Well, thanks, Laura. This has been brilliant. Let me just pause the recording.